Telomere replication by the Allelians. Before we explain what a telomere is, let's first demonstrate the problem it addresses. When DNA is synthesized, RNA primers attach to the parental strand, and DNA polymerase III fills in the gaps between the primers on the newly synthesized strand. The RNA primers are then removed, and DNA polymerase I fills the gaps left behind by the primers, moving from the 3' to the 5' end. The end of the strand, however, has no 3' hydroxyl for DNA polymerase I to build off of, leaving the newly synthesized strand slightly shorter than the original.
The end of the chromosome contains a G-rich series of repeats called a telomere, specific to each organism. Telomerase recognizes the tip of the telomere and attaches to the 3' hydroxyl. Telomerase is an enzyme that contains an RNA template to synthesize the 3' to the 5' direction, adding more repeats. This can sometimes be hundreds of base pairs to the parental strand. This is called reverse transcription because an RNA template is being used to synthesize a DNA strand. An RNA primer then attaches to the 3' end. DNA polymerase alpha, which has a DNA primase subunit, completes synthesis of the lagging strand. DNA ligase then seals the remaining gap. Even though the lagging strand is still shorter than the parental, it is the same length, if not longer than the original parental strand, meaning that all the original information on the chromosome is copied. These G-rich tails are capable of forming loops by doubling back on themselves and creating G-to-G hydrogen bonding. That, with added specific proteins, makes what is called a G-quartet or T-loop at the end of the chromosome to make them resistant to nucleus digestion and DNA fusions. However, most eukaryotic somatic cells do not contain
telomerase.
Each replication, the telomere is shortened. This limits the number of replications each cell can do, thus limiting longevity of tissues, organs, and ultimately the organism itself. Stem cells and malignant cells are known to contain telomerase, and it is thought to be the reason these cells are immortal. With our knowledge on telomerase, it is now being theorized and tested that longevity of organisms can be increased with the introduction of telomerase to somatic cells and the removal of telomerase from cancer cells to prevent their infinite growth.
Let's go! Let's go!